Thanks for the invitation to come and present at this, what's turned out to be a fantastic symposium. So I'm going to be talking about arsenic and non-alcoholic fatty liver disease. And if you were here this morning, you saw some data that Gavin showed on this topic. And he really pioneered this idea and generated some of the very first data showing the link between arsenic and the progression of NAFLD. And what we're doing in my laboratory is using some omics approaches to generate novel hypotheses. And so to give you a preview of my talk, I'm going to show you some of the data we have from some metabolomic studies that we did with Sean Zhang here in the Department of Chemistry. Then I'll show some proteomics data that we did with Mike Merchant in the Department of Medicine. And then I'll show you some, one of the novel hypotheses that we've generated out of this and some of the data that we have in support of that and some of the questions that that hypothesis raised. So first of all, we've seen different versions of this now. The CDC has now posted the latest data on prevalence of obesity in the country by state. And I thought this is a good one this year. They've included a little stippled green bar color, stippled green, for the 15 to 20% prevalence, although they didn't get to use that color. But they put that on there because that's their goal. They want all the states to be down about 15%. And so they're optimistic that one day they'll be able to use that color. But they have added, unfortunately, since the last time they've made one of these maps, an even higher category now of greater than 35% prevalence of obesity. And so if you add in overweight people, BMI over 25, that's another third of the population. And so it's a problem that is persisting. And the reason that we care about this, of course, there's a lot of health effects that will stem from being obese or overweight. And the one that we think about in the lab is this non-alcoholic fatty liver disease, where it's a spectrum of disorders, really starting with steatosis and getting progressively worse to steatohepatitis, fibrosis, and cirrhosis. And so one of the interesting things about it is even though we have so many obese people, only 10 or 20% of those people will actually progress from simple steatosis to NASH. And so that really tells us that there's something else that's going on, and we've heard a lot about the two-hit hypothesis, and I'm not going to be any different than that. I'm proposing the same thing, that the fatty liver that in this case is generated from consumption of too many calories in the typical Western diet, then acts as a first hit that now sensitizes the liver to second hits. And we've heard of a variety of different types of second hits that one can have. And I think it's interesting. I've heard the hits move around, which one's the first, which one's the second. And even Dr. Pro and Dr. Hine were talking about phase 1 and phase 2 metabolism, which comes first. It's sort of the same problem, I think. But there are two hits. I think we can all agree maybe the order is a matter of some speculation. But so the general working hypothesis in the lab is that environmental arsenic is, in fact, a second hit when we're talking about diet-induced fatty liver disease. And so what is arsenic? I think, fortunately, I get to go before you, so I get to introduce the concept a little bit. And so arsenic is an element that is found in the Earth's crust. It was really identified first as a public health problem in places like Bangladesh, where there's high concentrations of arsenic in the ground, and where at one point there was a lot of waterborne diseases that were causing lots of problems with infant mortality. And those problems were solved by drilling a bunch of tube wells around the country and getting rid of the surface water where all the parasites were and going into the ground. But 20 or 30 years later, we discovered that the arsenic in the ground was leaching into those water supplies and causing lots of problems, particularly cancers. But in this country, and here's one of these tube well pumps that has been painted red, and that's a great public health solution there. Once you find a well that has arsenic in it, you paint it red, and then so presumably people won't use it for consumption. But in this country, we don't have those really high concentrations of arsenic. We turn on the tap here in Louisville. We're blessed with very clean water, some of the best water in the country, I'm told, and and I like the water. But you know, is this a type of thing? So we don't have the problems with the Blackfoot disease and the various cancers that they have in these areas of high arsenic concentrations. But I want to point out that we do have arsenic in this country, and this is from the U.S. Geological Survey, a map of municipal water supplies. And I know you probably can't read the legend here, but the orange and the red dots are all water supplies that exceed the EPA and the World Health Organization limit of 10 parts per billion of arsenic in the water. And so the red dots have greater than 50. And so there's certainly parts of the country where there's greater amounts of arsenic, and where people are actually drinking water with some arsenic in it. And if I overlay that first graph, or first map showing obesity, you can point out some areas even in this country, like in Texas and in Michigan, where you have high incidences of both obesity and relatively high levels of arsenic in the drinking water. And so we think that this is going to be a relevant concern even in developed countries like the United States. And so going back to this two-hit model of steatohepatitis, where the first hit will generate the fatty liver, the second hit then makes it progress on to NASH or steatohepatitis. And the model that we're using is the model that was pioneered by Dr. Artiel, where the mice are fed a Western diet that's high in saturated fat, not quite as high as the previous speaker, but 42% of the calories, and also has cholesterol in it. And then the second hit that we're using is to put inorganic arsenic or trivalent arsenic or arsenite in the drinking water. And these mice are co-exposed into both the diet and the arsenic-containing drinking water for 10 weeks. And Gavin had shown that when you do this, just giving the high-fat diet, you see an increase in body weight. They gain weight, and their livers get really fatty. The liver weight to body weight increases, and you can see increases in the hepatic lipids, triglycerides within the liver, and circulating cholesterol and hepatic cholesterol all go up. But when arsenic is added on top of that, you see increases in these inflammatory markers. You see the increases in AST and ALT and hepatic expression of TNF and PI1, as Gavin talked about, and decreases in the anti-inflammatory IL-10. That's associated with F480 staining, the marker of the Kupffer cell activation. So we see there's definitely an increase in inflammation when you add arsenic on top of this high-fat diet. And so one of the things we wanted to find out is why. So the first approach that we used was a metabolomics approach. And the experimental design was to take both a low-fat diet and a high-fat diet, and to include either arsenic-containing drinking water or just regular tap water. And both the diets and the water supply here have undetectable concentrations of arsenic. We then extracted the livers from the proteins from the liver and did a GC-GC mass spec approach to get the metabolome from these 4 different groups. And the algorithm that we used to sort of identify the metabolites was to collect all these spectra and to use the factory-contained Chromatoph ID software to get a list of potential metabolite IDs, and then go back and actually examine the retention times on the 2 GC columns and discard all the ones that don't make any sense. And then we're left with a shorter list that we can use, apply the statistics and find the list of metabolites that are actually changing among the 2 groups. And then the interesting metabolites, we can go back and verify the identification by injection of authentic standards and comparison. So this is just an example of the type of separation we get with the 2 chromatography columns. On the x-axis, we get the retention time on the first column, and the y-axis is the retention time on the second column. And this is just the total ion current. You can see you get a nice separation of metabolites, which increases our identification using the mass spec. So this is the statistically different metabolites, and comparing a low-fat diet and a high-fat diet. This does not include the arsenic exposure. And you can see that there's an increase in several amino acids, including the sulfur-containing amino acids, methionine, cysteine, and the cysteine metabolite taurine. We also see, not surprisingly, increases in a couple of saturated fatty acids, pentadecanoic and tetradecanoic acid, because we're giving these a high saturated fat diet. But this isn't the comparison that we really are interested in. We're interested in the effects of arsenic on top of the high-fat diet. And so this graph shows the metabolites that we found that were different between those two groups. And again, we see several amino acids and fatty acids changing. There was an increase in citrate, lysine, and cysteine, and then there was a decrease in glycine and the acidic amino acids, glutamate and aspartate, and a couple of the short-chain butyrate metabolites, as well as, interestingly, a decrease in one of the saturated fatty acids. But I want to draw your attention here to the three amino acids, cysteine, glycine, and glutamate. You may recognize those as the components of glutathione. And just to remind you what's going on, so cysteine is glutathione is synthesized in a two-step process. For the first step, the gamma-glutamyl cysteine lysate, I got that all wrong, didn't I? Gamma-glutamyl cysteine ligase takes cysteine plus glutamate, and then glutathione synthase makes glutathione by adding glycine to that. And so what we actually saw was a decrease in glutamate and glycine, which would suggest maybe an increased flux through this pathway, but we saw an increase in cysteine. And so that led us to thinking about this cysteine glutamate antiporter, which brings cysteine in and exports glutamate. And so that may be, this is an indication that there's a perturbation in this thiol regulatory redox system. And I'm going to come back to that, I think, again at the end of the talk. But first I wanted to, going backwards. So to conclude the metabolomics portion, we saw that arsenic increases inflammatory liver injury, but not in the low-fat diet mice. This concentration of arsenic didn't have any effect on the liver. And another thing is that the changes that we see with arsenic in the high-fat diet are very subtle, but they're reproducible. And I think that the patterns that we see that are changing can tell us something about the potential mechanisms. And I talked a little bit about the glutathione, but there's also the decrease in glycine. Glycine is actually an anti-inflammatory amino acid when it's added to the diet. We don't know if the converse is true. The same is true with some of the short-chain fatty acids like Dr. McLean was talking about this morning. And the other thing is that we only did one type of extraction and one platform for identification. So we're by no means seeing all of the potential metabolites that were different. We're only seeing the ones that were available to be seen by the approach that we used. And so that additional methods might give a better coverage of the metabolome. So now I want to talk about the proteomics approach that we were using. And this was the same experimental design with the low-fat diet and the high-fat diet with and without arsenic. Except this time now the proteins that were extracted from the liver were subjected to an LC-MS approach. And using this approach, we found more than 1,800 unique proteins based on their peptide sequences. And 386 of those proteins were greater than or equal to twofold different when comparing just the high-fat and the high-fat plus arsenic groups. Now, we plugged all those numbers into the Ingenuity IPA analysis, and it identified a potential or a list of potential upstream regulators, and the top one was a transcription factor called HNF4 alpha. 34 of the 386 targets that were different were identified as being potential HNF4 targets by this pathway. And of those, 82% of them, or 28, were all decreased in the high-fat diet plus arsenic group compared to the high-fat diet group. So what is HNF4 alpha? Dr. McLean actually introduced it this morning when he was talking about alcoholic liver disease and some work that Dr. Zoe had done showing that the alcohol can inhibit the DNA binding activity of this transcription factor without changing its expression level. And so just to give you a little more information on it, since it also appears to be playing a role in this non-alcoholic steatohepatitis model, it's a homodimeric transcription factor, and this picture is from a recent review by Chandra, where each monomer is a different color, the purple and the yellow, the orange and the yellow. And DNA is shown here, it's sort of moving into the plane of the screen. And the DNA binding domains of each monomer sort of are sandwiched around the DNA. But the point I wanted to draw your attention to is that these DNA binding domains have two little circles in them. And those are the two zinc atoms per monomer that stabilize the overall structure of the DNA binding domain. And so we went back to the livers from these mice to see if we could see any changes in HNF4. It's not a very abundant protein, being a transcription factor, so it didn't show up in the proteomics analysis. But when we did a Western blot for HNF4, we actually saw an increase in the total amount of protein in the low-fat diet when arsenic was given, but that did not correspond to an increase in DNA binding activity that we measured with a DNA protein ELISA. In a high-fat diet, the protein levels weren't affected by arsenic at all, but we saw about 30% drop in the DNA binding activity. And that was very similar to the magnitude of the drop that Dr. McLean showed earlier today in the livers of alcoholic mice. And so this is a cartoon of just the DNA binding domain of one of the monomers of HNF4 alpha, showing the two zinc fingers. And if you'll recall, the zincs can be coordinated by either histidines or cysteines, but in this case, all the coordinating residues are cysteines. And there are other modifiable groups in here. We see a number of arginines, which can be methylated potentially, and lysine, arginines, which can be methylated, and lysines, which can be acetylated, and there's serines, threonines, and tyrosines, which of course we all know can be phosphorylated. And some of these residues have actually been studied, and this serine in between the two zinc fingers is actually fairly well conserved among a large number of these nuclear receptor superfamily members, and phosphorylation at this site can control nuclear translocation of a lot of these. So one of our thoughts is that because the protein levels aren't changing, that we're somehow modifying this DNA binding domain, and that could happen in a number of different ways. We could be changing one of these post-translational modifications, or we could be, in fact, dislodging the zinc and losing the overall structure. And that could happen because arsenic, first of all, likes to bind to thiols. That's been known for a long time. Or we could be oxidizing the cysteines, in which case, if they're in disulfide bonds, they can't bind to the zinc either. And so, to wrap up the proteomics part, I would say that that we see a prediction based on the proteomics data of an upstream regulator that we couldn't see, and when we went back, we could verify that experimentally, and that the effect appeared to be manifested at the post-translational level. So that got me to thinking about some old data that I had generated shortly after leaving Dean Jones' lab, because I spent my time there thinking a lot about the protein thyrodoxin. And so some of the data that I did when I was studying thyrodoxin may have bearing on this new idea with HNF4. And so this little diagram shows a protein that has two cysteines in it, the two thiols, that could be potentially oxidized. And there's some evidence that zinc fingers may be particularly susceptible to this type of modification because the cysteines are in the thiolate form, and therefore readily oxidized. And so if a reactive oxygen species comes and oxidizes the protein, it has to be reduced before it can be restored to its native state. And thioredoxin is the protein that is responsible for that. And through a thiol disulfide exchange, the disulfide bond is just transferred to the thioredoxin, which then is reduced by a protein called thioredoxin reductase. And over a decade ago, Stiblow and Thomas showed that several arsenic metabolites, particularly a monomethylated metabolite of arsenic, could inhibit the thioredoxin reductase. But at that time, they didn't have a way to measure any of these other processes, or particularly where the thioredoxin was being oxidized. And having done that with Dean, I applied that then to this. And these are some studies that I did a few years ago. And we learned something really interesting besides this. Now, this is a HeLa cell experiment. We simply added monomethyl arsenic to the HeLa cells. And here I want to make the point of the concentrations that we used. So this is thioredoxin reductase activity. And when we added 3 micromolar, we didn't have any effect. If we tripled that concentration to 10 micromolar, we saw loss of over 90% of the thioredoxin reductase activity. So I'd argue that we were putting in just enough monomethyl arsenic to get complete or near complete inhibition of this enzyme. And as a control, we were using a rheumatoid arthritis drug, orothioglucose, which is well known to inhibit thyrodoxin reductase. And here, I would point out that we added very high concentrations. 1 millimolar orothioglucose knocked out 90% of the thyrodoxin reductase, and then we put 10 times more than that. And it'll become obvious on the next slide, I think, why I'm making a big deal about these concentrations. So we measured the thyrodoxin redox state, and it tells you what the proportion of the thyrodoxin is in the oxidized form or the reduced form. And what was surprising was that orothioglucose had no effect on the redox state of thyrodoxin. Even when we put in 10 times more than was necessary to inhibit. But monomethyl arsenic, in stark contrast, putting in just enough to inhibit the reductase resulted in 70% oxidation of thyrodoxin. And you can see that under normal conditions, the vast majority of thyrodoxin is maintained in the reduced state. So this was really unusual. We actually confirmed with thyrodoxin reductase knockdown that it behaves like the orothioglucose. So simply knocking out that reductase is not enough to oxidize thyrodoxin. It turns out you also have to generate reactive oxygen species. And we measured that in this experiment with DCF fluorescence, which reacts primarily with hydrogen peroxide. But we see no increase in DCF fluorescence with the orothioglucose, but a big increase with the concentration of monomethyl arsenic that was also oxidizing thyrodoxin. And so coming back to this cartoon, I would say that at least with monomethyl arsenic, we are not only inhibiting the reductase, as Thomas and Stiblow showed, but we're also generating ROS, and others have shown that that's most likely through an NADPH oxidase mechanism. Aaron Barchowski's work that I think Gavin was talking about, they showed that as well. And so we're actually coming at this thing from both directions, putting an undue pressure on the thiol redox state of the cell, and now we go back to the metabolomics data showing that there was a potential perturbation of the glutathione system, and you can see that it's a real potential problem in terms of thiols, particularly like zinc fingers that reside within HNF4 alpha. And so what we're pursuing in the lab then is the mechanisms by which arsenic could be inhibiting this. Obviously, one mechanism is that it's displacing the arsenic from displacing the zinc from the sites, either by swapping out or through oxidation of those cysteines. Or that could be done in combination with this overall disruption of the glutathione or the thyrodoxin systems. But there's still the possibility that some of these other post-translational modifications are playing a role. We know that methylation of arsenic is a main way for metabolizing it, and there's some evidence that acetylation and phosphorylation cascades can be disrupted by arsenic exposure. And so I'd like to end by thanking all my collaborators, particularly Mike Merchant, who did the proteomics, and Shang Zhang and his group, who did the metabolomics, and of course, Gavin Artiel and the people in his lab who've been very helpful. And finally, I'd like to thank the NIEHS for supporting not just these studies, but also this symposium that Matt has organized. Thank you.
So we just came from a meeting where Jim Liu was actually demonstrating the first proposal you said as other mechanism, where arsenic actually replaces zinc and zinc finger binding, and because of the valence status, it only can bind 3 out of 4 cysteines, therefore destabilizing the DNA binding element. So what he proposed then was that arsenic essentially replicates zinc deficiency. Do you know anything about zinc deficiency in this system?
I do, and Jin Liu's been doing a lot of stuff with PARP in particular, and he's been characterizing the cysteine and histidine coordinating sites, and certainly it's not all arsenics are created the same. Arsenite and methyl arsenic have different preferences for these bindings. But in terms of HNF4, a zinc chelator will actually inhibit its DNA binding activity. So if you strip the zinc out, then you can inhibit the activity. And with alcohol, where you lose activity, and in the studies that Dr. McLean referred to, if you give supplemental zinc, you can restore the activity. But it's not clear whether that's through reintroduction of zinc or blocking its loss in the first place, because it's given not therapeutically, but preventively, and zinc can act as an antioxidant, and so it may just be preserving those zinc fingers. So that's a good point, and it's certainly one of the things that we have to consider. Yes?
So the zinc and alcohol partially restores it, almost does. So for arsenic, if you wanted to totally fix things, you would give zinc and a glutathione prodrug together, is that right?
Well, that's what I would do, yes.
So that should be your therapy for Bangladesh.
Okay.
Very nice talk. In the scheme with the thyroidoxine, you were suggesting maybe the zinc thiolates are also oxidized by the system. Are there data showing that the zinc thiolates can be directly reduced by thyroidoxine, or is this an indirect by protecting the thyroidoxine reduced state? That by default, the zinc thiolates should also be protected too.
Well, those are absolutely key questions, and I don't think there's not a lot of data on restoring these zinc fingers once they become oxidized.
Right.
And so certainly there's more than one mechanism to restore those, and thyroidoxine is one of those, and glutathione-dependent systems are another, and we're just going to have to go in and look to find out what's going on. Yes.
In your studies, did you measure reducing equivalent ratios with, for example, NADPH to NADPH or NAD to NADH ratio?
We did not.
Just out of curiosity, with the link to thyroidoxin, you might want to consider reducing the NADPH levels because of the steatotic pathways.
That's a great idea.
Arsenic didn't affect GSH in the original study.
No.
One last question. I really enjoyed the talk. So, have you considered the possibility, the alternative possibility, or in addition to, because you're basing most of what's going on here to actual post-translational modifications on HNF4 alpha, right? Itself.
Right.
So, A, have you been able to show that with arsenic you have conformational changes in the protein, or B, could it be possible that in addition or instead of a post-translational modification that arsenic might be changing the consensus sequence in the DNA that binds for HNA4 alpha?
That's an excellent point. And that's one of the things that we have proposed to do is to consider if we had simply changed arsenic for zinc, have we changed the types of promoter elements with which it interacts? Because there's a lot of play in an HNF4 response element. And so there's lots of different types of sites will bind. And so that's one of the ideas that we plan to test is in the presence of arsenic will now bind to a different subset of things.
Do you know if the consensus sequence is specific for HNF4 alpha, or does it bind other transcription factors?
It will bind to others.
Huh?
Yeah, no, it's not specific. There is
So if it does, you
overlap.
could look to see if there's a global decrease in recognition of that consensus sequence or other things that bind in there. So that might be telling you that arsenic is affecting, that DNA domain too.
Great. Thank you very much.
Okay, thanks, Bert.
Thanks.